I am Dr.Ashok Kumar, Department of Zoology and Industrial Fish and Fisheries, Ganga Singh College, Chapra, running under JP University, Chapra. Today I am going to switch on the topic cryopreservation of gamete. Cryopreservation of gamete, it has become a new frontier in the field of aquaculture. It means this cryopreservation of male gamete has become possible till date. No, it has not been possible for the scientists to preserve the female gamete. Hence, only male gametes till date, only male processes have been have been prepared for preservation of male gamete or spermatozoa for a long period, for 5 years, 10 years, up to 10 years, but it depends on the raw milt. So for today, for aquaculture, for selective breeding, for hybridization, for commercial seed production, this has come forward. This process of cryogenic preservation. Cryogenic means long-term preservation. So long-term preservation of milt has become milt is possible because only a spawn for under controlled condition. We can also get the spawn containing ovum by stripping the belly of the female brood stock, and which comes, and in a dish it is collected, and these cryogenically preserved spermatozoa or milt, these are poured, and then with the help of feather it is mixed and fertilization takes place, and after fertilization they develop in hatchling, and in this way, hence this cryogenic preservation, that is long-term preservation of milt or of milt containing spermatozoa, male gamete, has become possible, and for that preservation certain steps are involved in this preservation. First thing is, this preservation is of two types. Preservation of two types. First is non-cryogenic, that is short-term preservation, and cryogenic preservation means long-term preservation. So this is the difference. So for selected sample we keep alive. For selected sample we keep alive the spermatozoa in the milt. Now the first step is preparation of extender. This extender, it is the first step, preparation of extender. It is used in both types of preservation, whether it is cryogenic or non-cryogenic. It is prepared. So some of the workable extenders are as follows. First, NaCl 400 mg, CaCl 23 mg, KCl 38 mg, NaH2 sodium bicarbonate, and Na sodium bicarbonate 100 mg, NH2PO4 48 mg, MgSO4 23 mg. In 100 ml of distilled water, these salts are dissolved and second is, so it is first type of extender. Second is the same. Second is NaCl 600. There is difference in weight. Second is, in first NaCl was 400, but in second it is 600. So second B, it is NaCl is 600 milligram, calcium chloride 23 mg milligram, KCl 38 milligram as in first, sodium bicarbonate 100 mg, NaHCO3 100 mg, NH2PO4 that is 41 mg, MgSO4 magnesium sulfate it is 23 mg. These salts are dissolved in 100 ml of distilled water. These are for during cryopreservation. Third is NaCl, CaCl2. C is NaCl. Now NaCl we have seen in the above extenders the sodium chloride fluctuates. Here also in third it is 700. NaCl is 750 ml. Mg 750 mg, calcium chloride 20 mg, potassium chloride 20 mg, sodium bicarbonate NaHCO3 is 20 mg. In these salts are dissolved in 100 ml of distilled water. So in out of the three, this extender means the last extender where NaCl 750 mg, CaCl2 20 mg and potassium chloride 20 mg, sodium bicarbonate 20 mg. These are dissolved in 100 ml of distilled water. It is used in non-cryogenic C. This last one is used in non-cryopreservation, means not long-term preservation. It is C and the A and B, where NaCl 400 mg, CaCl2 23 mg, the same CaCl2, KCl2, NaHCO3, NaHCO3, these ammonium NH3CO3 and NH2PO4, these are they similar, almost similar, and in A and B are for cryogenic preservation and C is for non-cryogenic preservation. Now now non-cryogenic preservation, it is very short-term preservation. That is the milt, raw milt containing spermatozoa, they are preserved for 3 to 4 days. Hence it is non-cryogenic. It is not long-term. It is non-cryogenic preservation and 3 to 4 days, for 3 to 4 days non-cryogenic. And the milt sample is diluted with extender stored in a thermocol chamber with ice. The extender, what we have prepared in non-cryogenic, that is the non-cryogenic preservation, that is C-type extender, and then that and milt sample is first diluted with extender. The milt sample is diluted. In extenders, the samples are diluted. In extenders, the milt sample are diluted in extenders and stored in, and the milt sample are stored in thermocol chamber, that is, and with ice or in refrigerator, so that the temperature should not rise. That should be in preserved condition in refrigerator. Extender C is found more easy and effective. Extender C have been found more easy and effective for non-cryogenic preservation. For such non-cryogenic preservation, milled samples are brought to gradually milled samples are brought to room temperature before inseminating the egg sample for better result, because we stripe the abdomen of the female brood stock, and that after stripping, the spawn comes in the dish, and in that dish the milled sample which has been which was kept in refrigerator in extender, and that is we take it from outside take it from the refrigerator, and later on we bring the milled sample on room temperature, and then we pour it on the pour over the spawn containing ova, and fertilization takes place after mixing with the feather. Fertilization takes place after mixing with the feather. Now, it was all about the non-cryogenic preservation. Now, cryogenic preservation, that is long-term preservation. It is spermatozoa can be stored for years together in liquid nitrogen. In liquid nitrogen means nitrogen generally in air remains in gas form, but at very cool temperature is kept cooled, and this nitrogen gas becomes liquid and are kept in thermoflask, liquid night, and the temperature in minus 190 degrees Celsius, at minus 190 degrees Celsius, where at this temperature all the biochemical activity of living cell ceases. That is, the biochemical activities of the living cell means spermatozoa that ceases, that do not no activity is found there, and it ceases. Further, the milk is diluted in extender, as I had already explained, that it is milk is diluted in extender and mixed with cryoprotectant. Cryoprotectant means what is cryoprotectant? Cryoprotectant, it is a chemical which protects the cell from chilling injury. From chilling, generally from chilling, as we keep some ice in our hand, a chilling injury, our skin shrinks. So from chilling injury, this cryoprotectant protect the spermatozoa, protect the cell membrane of the spermatozoa. This cryoprotectant, which is they protect the cell from chilling injury during the course of freezing. So at minus 190 degrees Celsius, that is the nitrogen becomes liquid, and it is kept, sperm is kept there, and from chilling injury, cryoprotectant, we use cryoprotectant, that is glycerol and DMSO, dimethyl sulfoxide, that is DMSO, that is cryoprotectant. Now The extenders now protocol for cryopreservation are as follows. The protocol for cryopreservation is first is preparation of extenders, which I had already mentioned, that different salts were used. They should be measured, weighed accurately, and they should be dissolved in 100 ml. In 100 ml in distilled water or double distilled water, and are the so they accurately, and that should be those salts should be in solution form in distilled water, and the desirable pH of that salt solution should be 7.2, 7.2 to 7.3. It is the desirable pH of the solution, kept under refrigeration, kept under refrigerator, and can be used safely for a month. This type of extender can be workable only for 1 month. They cannot withstand, and they can become spoiled after 1 month. So up to 1 month they are working. Now the second is first is preparation of extender, second is preparation of diluent. Diluent means diluent. It means two things are mixed, DMSO and glycerol. So glycerol and DMSO. So first is the cryoprotectants used in present protocol are DMSO and a combination of DMSO and glycerol, dimethyl sulfonoxide and glycerol. Two things are here, and diluents are hence it is called diluent, DMSO and glycerol. The cryoprotectant is mixed directly in the milled sample. The cryoprotectant is not mixed. Cryoprotectant is not mixed directly into the milled sample. It is mixed first with extender in the required ratio. First, we had already prepared extender. So this diluent is not directly mixed with the spermatozoa, but it is mixed with this diluent or cryoprotectant is mixed with the diluent for the first initial. Diluent is mixed with the extender in the required ratio, and then it is kept into the refrigerator due to its exothermal reaction. Now it is kept in refrigerator. The ratio of external and cryoprotectant in different diluents are as follows. The ratio of extender and cryoprotectant. First we had prepared extender, and second it is cryoprotectant. So the ratio should be in the as I am going to mention. First diluent is prepared glycerol 10 ml, DMSO 5 ml, and extender. Now these diluents are mixed with the extender. So 10 ml glycerol, DMSO 5 ml, and extender 90 ml. In this ratio, extender 90 ml and 10 ml glycerol, DMSO 5 ml. Second is glycerol, again 10 ml, DMSO 8 ml, and extender 90 ml. And third is DMSO 15 ml and extender 65 ml. So without glycerol, only dimethyl sulfoxide, that is 15 ml, and extender 65 ml. The working ratio of milled and diluent is 1:4. The working ratio of milled and diluent is 1 into 4. Milk should be 1 ml and diluent should be 4 ml. So now in the equilibration, it will I will narrate it in next class, our next class. Thank you, thank you, thank you.